In this video, we'll be looking at Mendelian Genetics Part 2, specifically looking at things that don't always follow the rules of dominant, recessive, and some of the different things that we talked in the previous video. This first concept is the concept of incomplete dominance. When you get confused on these, just look at the name. It tells you what's going on. There's dominance, and we understand the idea of dominance, right? Dominant always expresses itself. However, this one's incomplete dominance, so it's not completely expressing itself. So I'll give you an example. And this incomplete dominance always has to do with more than one dominant trait. That's important. So in these flowers, whatever these flowers are typically referred to as snapdragons, red and white are both dominant. So when you mix them together, let's say you have a homozygous red and a homozygous white that are mixed together, you're not going to get, since they're both dominant, they're both wanting to express themselves. And so they are both incompletely expressed. Neither one of them completely dominate, hence incomplete dominance. So red isn't quite red, white isn't quite white. They're pink. And so the heterozygous condition is pink. Well, if you mix two pinks together, what would you expect? You'd expect to see, right, some red, some white, and some pink also. Same sort of idea, but just two dominant traits. Now, what about, and this incomplete, that's different from complete dominance again, which we already talked about. What about in an instance when you have two alleles that are both dominant, but they share dominance? This is called codominant. So if you want to go back to this one real quick before we talk about blood types, in a codominant situation, you say you have more than one dominant allele. This is where both of them are expressed and they share dominance. And so you would have a red and white flower as opposed to a pink flower. In our own bodies, we have codominance expressed in our blood typing. Well, not necessarily your body or my body, but somebody's have this. All right, so I'll explain what that means. Type A blood is dominant. Type B blood is dominant. If you have both, you don't have a blended version of them. You don't have type C blood. That would be incomplete dominance. Instead, you have type AB blood. You show both characteristics of type A and type B blood. Type O blood, on the other hand, is neither. Type O is a recessive trait, and it doesn't show either one of those. So it's possible that you could be type OO blood, meaning that you have two O alleles. It's also possible that you could be, so like this type A blood person here, they're type A, but they carry one recessive allele, right? And so they're like, if you wanted to think of it this way, they're type AO, right? They only show A because dominant only shows, but they can pass the O allele. And same with the B. So O has neither. Now, this stuff about anti-A and anti-B and so forth, basically type A blood doesn't like type B blood. So if it recognizes it, it forms antibodies against it. Type B blood is the same with type A. So if you're type AB blood, it would be really bad if you had anti-A or anti-B in you. So you have neither. And so someone who's type AB blood can actually accept any kind of blood. AB blood is called the universal recipient because of that. Type O blood, on the other hand, has anti-A and anti-B blood. But here's the catch. Type O blood is not recognized as anything. And so it doesn't matter what type of blood you have, you can accept type O blood. And so typically type O blood is called the universal donor because it doesn't have anything that makes it special, that makes it look like any other type of blood, so that it is able to donate that blood to any other type. A different kind of trait that has to do with this variance of basic Mendelian genetics is called pleiotropy. And pleiotropy is where you have one gene that can affect multiple phenotypes. Sickle cell anemia is a great example of this. if you have sickle cell, there's one gene that is mutated in your genome. However, that gene affects multiple phenotypes. It affects the shape of blood cells. It affects those blood cells' ability to carry oxygen. It affects those blood cells' resistance to certain pathogens like malaria. People who have sickle cell, for instance, are resistant to malaria, and we'll talk about that in another video, actually. And so there's multiple phenotypes that are affected by that one gene. Another idea that is different than just basic Mendelian genetics is this idea of epistasis. Epistasis is where a gene at one location can affect a gene at another location. So similar to pleiotropy, but very different in that this hair is the best way to explain this idea of epistasis. So you think about someone's hair. Picture in your mind someone's hair or your own hair. There are numerous phenotypes that are coming together to produce the thing that you see. If someone has brown hair that's thick and curly, it's going to be like almost afro-ish, But if someone has brown hair that is thin and straight, it's going to look completely different. And so all of those genes are affecting one another in much different ways. All right. They are affected. So color, shape, thickness, texture, all of those things come together to create this certain sort of trait, if that makes sense. Let's see. Next thing is polygenic inheritance. Polygenic inheritance is where, so look at the word polygenic, more than one gene. All right. And so this is where you have multiple genes that affect one single phenotype. Skin color is the best example of this. There's multiple genes that code for skin color. And there's this idea, let's see if I have another picture of this. I don't. So there's this idea of something called quantitative characters. When you hear the word quantitative, you immediately think of like counting, right? And so this person has multiple genes across the spectrum of their skin color. And this is just showing 3, actually. And so in this particular instance, all 3 genes are not expressing, right? And so there is this one type of color. Over here, all 3 genes are homozygous in their expression. And so there's one other extreme. And all in between there are different variations of that color. In this particular version, only one bit of A is turned on and B and C are turned off. And so that adds up. That's where you get this quantification difference. That adds up differently than, say, this one. And so in reality, there's a lot more than 3 genes that code for skin color. And so as the effects of those genes add up or quantify, you have a particular expression of that gene. So if you think about just think about the variation of skin colors in our own building. You line them up, you would get really pale to really dark and almost everything in between because of the way that those genes are inherited. And the last thing is something called multifactorial. This has to do with environmental conditions affecting a phenotype. And so, a very simple idea of this is just plants. And so in this particular example, you have, I forget the name, maybe hydrangeas, what that is. So you have a plant over there, and if you grow that plant in a certain sort of pH of soil, you can get it to be more pink than purple. And so in this case, it's not the gene that's causing the change in color, it's the environmental condition. Okay, that is a really plain thing to think about, but this brings up the whole nature versus nurture in organisms as well. You take a person, for instance, who has a certain set of certain set of circumstances in their life their genes are going to be expressed in a certain way versus a person who has the same maybe genetic makeup but has a different set of circumstances in their life. Twins, I mean, you just take, for instance, identical twins separated at birth. This is just an example. And you put one in one set of circumstances and the other in another set of circumstances, they're going to express different phenotypes because of the environmental conditions from which they grow. Something like, things like height that could just come from just food intake, intelligence, all of this stuff has to do with environmental conditions that aren't necessarily completely controlled by the DNA.